Hey all, so today we will discuss about dental caries. So as we know that our tooth structure is made up of inorganic component and the organic component. So inorganic component is mainly composed of hydroxyapatite crystals, which are basically the minerals, and the organic component is made up of the collagen fibers. So in dental caries, which is an irreversible process, the minerals which are present in the inorganic component will be demineralized. So we will say that there will be demineralization of the inorganic component and the organic component will be destructed. There will be destruction of the collagen fibers. So now we can easily understand the definition of the dental caries, which is an irreversible microbial disease of the calcified tissues of the teeth, characterized by demineralization of the inorganic part and destruction of the organic substance of the tooth, which often leads to cavitation. So the next topic is etiology of dental caries. So there are main three theories that we will discuss about. So the first one is Miller's chemicoparasitic theory, the second one is proteolysis theory, and the third one is proteolysis chelation theory. So the first one, that is Miller's chemicoparasitic theory, is also known as acidogenic theory. And this theory states that caries is caused by microorganisms which are present in the mouth. And this theory also states that dental decay is a chemicoparasitic process in which there are two stages. The stage one is decalcification, and the stage two is all about the dissolution. So initially, the enamel and dentine gets decalcified, all the calcium is leached out, and after that, the softened residue is basically dissolved, or dissolution of that residue takes place. He gave importance to microorganisms, carbohydrates, and acids. So, the microorganisms present in our mouth will act on the carbohydrates which are retained on our tooth structure, due to which the microorganisms will produce acids, and this acid will eventually dissolve our tooth structure. Now we will discuss role of each one by one. So the first one is role of carbohydrates, in which Miller incubated the tooth structure inside a mixture of saliva and carbohydrates. So he saw that there was decalcification of the tooth structure, whereas the same tooth structure, when kept inside a mixture of raw starches and saliva, then there was no such decalcification seen. Raw means basically uncooked. So the refined diets that we are having nowadays, or packaged food which are readily fermentable, are basically responsible for dental caries, or you can say reduced resistance toward dental caries. Now, according to Miller's, the carbohydrates were responsible for tooth decay. So, the bacteria which is causing dental caries, or cariogenic bacteria, are dietary in origin, not salivary. Nextly, the karyogenicity of dietary carbohydrates depends upon the frequency of ingestion. So, how many times in a day you are taking these carbohydrates. Secondly, the physical form, whether it is sticky or non-sticky. If it is sticky, it will be retained on a tooth structure for a longer period of time, and it will cause tooth decay. Thirdly, the chemical composition, whether it is fermentable sugar or a non-fermentable. If it is fermentable, then it will cause tooth decay. Fourthly, the route of administration, whether we are taking it orally or via any other route. If we are taking it orally, obviously tooth decay will take place. But if we are taking via another route such as IV, then there will be no such phenomenon seen. Then presence of other food items. So, as we have discussed that the cariogenic bacteria is dietary in origin and not salivary, because saliva contains negligible amount of sugar. Even if some amount of carbohydrate is present in the saliva, it is bound to the proteins, and it is not readily available for microbial degradation, or you can say fermentation. Now we will discuss about role of microorganisms. So Miller has demonstrated the presence of microorganisms within the dentinal tubules of the decayed portion of the tooth. He called them cocci and leptothrix. After that, J. Killian Clark has also isolated Streptococcus species. Other than that, Lactobacillus and Actinomyces species were also isolated from dental caries. So there are many studies demonstrating that certain type of or specific type of microorganisms causes specific type of caries. Although there are many doubts and disagreements to such specifics, still it is believed to be like that. Firstly, there are pit and fissure caries, which are caused by S mutans and lactobacillus. Secondly, smooth surface caries, which are caused by S mutants. Likewise, A viscosus and A nacelundi causes root surface caries, and deep dentinal caries are caused by lactobacilli, A nacelundi, and other filamentous rods. So it is believed that some of the microorganisms are responsible for initiation of dental caries, whereas others are responsible for progression of such caries. Now we will discuss about role of acids. So the microorganisms present in the oral cavity will ferment the sugar, or you can say the process of glycolysis start, due to which there is production of acids. So these acids basically harm our tooth structure. Now the microorganisms which produce only a single type of acid are known as homofermentative, such as streptococci and lactobacilli, which are producing mainly or 90% of lactic acid only. But in addition to the lactic acid, if two or more organic acids are produced, then these bacteria are known as heterofermentative. Now, the presence of acids is less significant, but the localization of these acids play an important role. So dental plaque play an important role in holding the acids in the oral cavity for a longer period of time. So after fermenting the monosaccharides and disaccharides by the microorganisms, there will be increased amount of acid production that will lead to greatest amount of fall in the pH in the dental plaque. Now, as we know that the microorganisms are fermenting, it means they are initiating the process of glycolysis only. So, one molecule of glucose will produce two molecules of lactic acid, as we have read in the glycolysis process. Secondly, the amount of lactic acid produced depends upon the type of bacteria. So, the acid production by S mutans is more than S sanguis. Secondly, the growth conditions. S mitis and actinomyces are homolactic in anaerobic conditions, whereas in presence of CO2, these are heterolactic. So, as we know that the Millers has given importance to bacteria, microorganisms, and the organic acids produced by these microorganisms. Still, there were some shortcomings. So, the first one is the predilection of specific sites on a tooth to dental caries. As we have read, there are some specific microorganisms which act on specific surfaces. Secondly, initiation of smooth surface caries. Thirdly, you have seen some individual who are caries free. So caries free population wasn't explained by him nicely. Fourthly or lastly, the phenomenon of arrested caries. So these were all the shortcomings of Miller's